Good evening, ma'am. Good evening, Agil.
Good evening, sir.
Good evening, ma'am. Good evening, Ajay.
Good evening, sir.
Good evening, Gana.
Sir, you can start, sir.
Yes, ma'am. Thank you. Good evening, my dear students. Today, we are going to discuss about the new chapter and also very interesting one. Don't skip this chapters because it is very, very important. Maximum question may arise, may asked from this portion only, this lesson only. That is molecular basis of inheritance and also principles of inheritance and variations. This is the topic name. Here we are going to discuss about the complete DNA structures, what is my genetics, alleles, all those things, monohybrid, dihybrid, incomplete, codominant, epistasis. Okay, that things we are going to discuss, and what are the sex-linked inheritance. Okay, I ask you before that. You all look like your parent, isn't it? How it will be? What will be the main important component or molecules. Anybody else? Ajay? Please. You are all look like our parent, isn't it? So what is the reason behind it? Why we are looking as a parent? We are having both a character from parents, isn't it? Why? Jaswant, Nagil, anybody else? One of the most important component is called a genetic material, isn't it? Genetic material. The genetic material is mostly, what is that? A major component, DNA as a genetic material, because it transfers the characteristics from one generation to another generation through chromosome, or what is that? A nucleotide. Okay. See here. So before that, we must know some basic term. What are the basic terms we are going to discuss about in this chapters? First one is called a gene. Very, very important one. What is meant by gene? It is nothing but the unit of inheritance of characters. Such type of, what is that? Unit is called as, what is that? Gene. Already you know well about what is meant by genetics. The science that deals with the study of gene is called as genetics. Okay. But gene is a unit of inheritance. This is also a very, very important question. Okay. Unit of inheritance of the characters. Okay. They may ask the question directly or assessment reasoning like that. So don't forget it. Here, the gene, the unit of inheritance, unit is nothing but the, what is that? Measuring. Okay. So the inheritance of the character is called as gene. Second one is called as alleles. Already known very well. What is the meaning of alleles? See here, that alternative form of gene having the same locus on chromosome is called alleles. Here, which one is called as alternative form of gene? Sometimes they may ask the question, that is called as alleles. Which one is the alternative form of gene? Alleles. Okay. It presents the same locus. Locus is nothing but the location of the chromosome, on chromosome. Take an example, any one of the chromosome. Okay. They take a small portion. That portion is nothing but the location. The location, it has a positive character as well as a negative character, otherwise called as a dominant character as well as a recessive character. So both are sometimes present the same location, that is called as locus. Okay. So this is very, very important one. First one is gene, the unit of inheritance of the characters. Second one is called as alleles. Alternative form of gene having same locus on chromosome is called as alleles. And third one is called as autosome. So autosome is, what is the meaning of autosome? Autosomes, see here, all chromosomes other than the sex chromosome is called as autosome. We are having 46 chromosome, isn't it? Here, how many pairs of chromosome present in it? 22 pair. What is that? 22 pairs. What is that? Autosome. Only one pair is a sex chromosome. See here, 46 chromosome is there. That is called as 23 pair of chromosomes. That is called as autosomes. Other than autosome is called as allosome. This is called as XY. It is denoted as a sex chromosome. Already we studied, isn't it? Sex chromosome means, but the female. Already sex determination already we studied. XX denoted as a female. XY denoted as the Y is the gene is present in it. That baby is male characters. That means boy. So this is a sex chromosome. So other than the autosome is called as allosome. Other than the allosome are called as autosomes. Okay, so many times they were asked how many pairs of autosomes present in it. How many pairs? 22 pairs. Okay, last one is called as allosome, otherwise called as sex chromosome is there. Okay, note down, this is very, very important. Another one is called as factors. Factors is also denoted by the some of the scientists named. In ancient ages, they discovered the factors. Unit of hereditary. Here, the first one is called a gene, unit of inheritance, which is responsible for inheritance and the appearance of characters. The same, similar to that of the gene. Okay, who stated the name, who gave the name gene. That's also very, very important one. And first, the factors, who stated the statement of factors, that particular gene is also otherwise called as factors. This is also very, very important. We will study in the historical aspects. Okay, when you are going to study the lesson deeply, that time we can study these things. Okay, factors. Factors is nothing but the gene. Both are having the same, what is it? Explanation. Don't forget it. Okay. See, go to the next slide, my dear students. Here, any doubt, my dear students, in this part? Gene, alleles, autosomes, and sex chromosomes, and factors. Okay, see that. Okay, go to the next one. Linkage. What is the meaning of linkage? It is a tendency of gene present on the same chromosome to inherit together. Tendency of genes present in the on the same chromosome. Already we discussed, is nothing but the locus. Suppose any one of the locus, it contain, what is that? Same chromosome to inherit together. Then only it will be express itself. Otherwise, it will not express. Such type of chromosome or called as, or gene called as, what is that? Linkage. That process is also linkage. We are all also one of the hybrid of our parent, isn't it? Male as well as a female gametes. It's nothing but linkage. Otherwise, it's called as a crossing over. Takes place in the meiosis cell division in sexual chromosome. Already we discussed that things also, isn't it? Linkage is also same, similar to that one. We will study it. What are the linkages? What are the types of linkages? And what are the examples? That things we are going to study in deep manner. Okay, go to the next one. Punnett square. Square is nothing but the shape, isn't it? It is a graphical representation of the calculate the probability of the all possible genotype of offspring in a genetic cross. In our bird, monohybrid cross, dihybrid cross, test cross, and back cross, incomplete, complete. Likewise, there are so many, what is that? Hybridizations also done by many scientists, isn't it? Finally, the genotypical possibilities, it's also calculated in a small, what is that? Geographical representation, small square only, that's all. This was first introduced by the name of the scientist, the Punnett. That's why they called Punnett square. Okay, see here the Punnett square. Once again, repeat for you. Punnett square, it is a graphical representation of the calculated probability of all possible genotype. What is meant by genotype? Genetical oriented. What is meant by phenotype? Physically. Okay, visual aspects. Suppose, take an example, monohybrid, height is an example, isn't it? Height, one is height, another character is dwarf. When we are seeing the genotype or phenotypical characteristics, it is a physical oriented, isn't it? Physical oriented means we can easily visible. Genotype we cannot able to visible. It can be a small calculation. Then only you will get the possibility of genotype. It may be like that, homozygous and heterozygous, and also homozygous dominant, heterozygous dominant, and homozygous recessive. We'll see one by one, isn't it? Punnett square is nothing but the graphical representation of the calculated the probability of all possible genotype of offspring in a genetic cross. Incomplete dominant. It is also another type. Okay, when one of two alleles of genes is incompletely dominant over the other allele is called as incomplete dominant. We will discuss what is my incomplete dominant. Suppose when you are going to cross a red flower with a white flower, what will be the F1 generations? Which one is the dominant character? That character will be expressed in the F1 generation, isn't it? But instead of dominant character, what happened, the in between white and red, when we are mixing, what happened? You will get the pink color. Okay. It is, what is that? It is expressed in the dominant character. Red is the dominant, but incomplete dominant. It not fulfill. Such type of crossing is called as incomplete dominant. Which one is an example? Mirabilis jalaba. Note it down. This is very, very important. Incomplete dominant also done by the one of the scientists. We will discuss. Mirabilis jalaba and also called as 4 o'clock plant. What is meant by 4 o'clock plant? The flower blooming at it when 4 o'clock only. That's why the name is called as 4 o'clock plant. Otherwise called as Mirabilis jalaba. They are having white and red color flowers. Sometimes in some of the other varieties also present in it in this category. There are so many examples also there. Okay, so this is the incomplete dominant. These are the terms. What are the terms which comes inside the lesson? Next one is mutation. Already we discussed. What is my mutation? Sudden heritable discontinuous variations of our organism due to the changes of gene and chromosome is called as mutation. Otherwise called as sudden heritable changes occurs on the chromosome or nucleotide or gene is called as what is a mutation. It may caused by the physical and chemical, biological. Already we studied in cancer, isn't it? Biology in human welfare. That time, what are the things we studied? Cancer is also one of the sudden changes occurs due to the chemical substance or physical substance or biological substances. Okay. What happened, as a result, what happened, the gene code will be changed. And also it's excreted from the one generation to another generation. Such reaction is called as mutation. Okay. That is very, very important. Okay. What are the next one? Mutagen. Already we discussed. What is by mutagen? The chemical or physical factor that induce the mutation. Which substances they can ability to induce the mutation. Such type of components are called mutagens. They may be a chemical mutagens. They may be a physical mutagens. Some of them are biological mutations also there. About carcinogenic viruses. The carcinogenic viruses comes under the heading known as what is a biological. Chemical, but there are so many chemical substances also there. Okay. Tobacco, there are 60 or 80 alkaloids present in it. Okay. Nicotine, okay, nornicotine, anabasine, so many things is there. They are all induce the cancer. Okay, that is chemical. Physical, and also one more is that UV radiation. Physical is nothing but the UV radiation. UV radiation sometimes they may induce the what is that? Cancer. Okay. That is also very, very complicated. UV radiation mostly, what is that? Suppose, take an example. Our atmosphere is protected by the, which component, anybody else? Our atmosphere protected by which component, which molecule? Anybody else? Agil? Ajay, Jaswant, Ritika, anybody else? Nobody answer me? Hello? Okay, anyhow. So what about the ozone layer? Ozone layer prevent the what is that? Transformation of the UV radiation from the sun to atmosphere. Okay, so this is very, very important. And next one is called as point mutations. What is my point? There is any one of the point. It is an analysis of a triad in a several generation of a family in humans. This is called as point mutation. Point mutation takes place in any particular area or locus or a nucleotide. That point is called as point mutation. The mutation takes place in a point. Okay, this is also transfer from the one generation to another generation. For example, it's a family in humans, several generations. So that is very, very important. Particular point, it will be expressed into so many generations. And hemophilia, what is by hemophilia? Is one of the disease caused by the recessive, sex-linked recessive gene. See here, hemophilia, sex-linked recessive disease, which shows its transmission from unaffected carrier female to some male progeny due to lack of blood clotting factors. Okay, hemophilia is nothing but the, what is a recessive, this is very, very much sex-linked recessive disease. First to note it down. How many factors is there? Blood clotting factors. How many factors, my dear students? Anybody else? Ajay, can you hear my voice, my dear? Hello? Okay. And then hemophilia is nothing but, you know what about it is a sex-linked recessive disease which shows its transmission from the unaffected carrier female to some male progeny due to the lack of blood clotting factors. There are 13 factors is there, and 3 factors is for, what is that? This syndrome. Okay, genetical. Klinefelter syndrome. This is also very, very important. These are all very, very important genetic disorders caused due to the additional copy of X chromosome of sex in male, resulting in a karyotype of 47 chromosomes. Note it down. This is very, very important one. Klinefelter syndromes. The chromosome number will be 47. Normally, we are having 46 only. Here, 47. Which one is added here? You know, X. See here, one X is also, it is now this chromosome. That's why we are getting which one? You know, 47. Okay. See here, XXY. So here the XY is a male having the female characteristics. This syndrome is also sometimes happen during the crossing over. These are all are called as what is it? Klinefelter syndrome. Male having female characteristics, upper part and also the lower part. And phenylketonuria, it is also another inborn error of metabolism. So note it down, this is very, very important. Phenylketonuria, inborn error of the metabolism, inherited as autosomal recessive triads. Affected individual lack an enzyme that convert amino acids phenylalanine into tyrosine. So this is very, very important. Which one is the lack of enzymes? Sometimes they may ask the questions. This is already so many times they were asked. Okay, so phenylketonuria. Okay, it is also one of the inborn error of metabolism, inherited as autosomal recessive. Autosomal already in our body. It's nothing but the 46 chromosome in our about autosomal. One is the sexual chromosome 23 power, isn't it? The affected individual lack an enzyme. Which enzyme convert? Which enzyme? Okay, this is very, very important. We are going to study deeply. Convert amino acid. Which amino acid responds? Phenylalanine, phenylketonuria, phenylalanine into tyrosine. That conversion of the phenylalanine into tyrosine is the conversion with the presence of the enzyme. Suppose the enzyme is lack, what happened? You know, the conversion is not possible. That phenylalanine excreted in the urine. That's why that condition is called as phenylketonuria, ketonuria, which comes out through the urine. Mental retardation occurs as a defect. This is very, very important. Phenylketonuria, so very dangerous. Okay, So this is also an important one. And sickle cell anemia, you know what about RBC, RBC, the normal shape of the RBC, how it will look like? Anybody else? Ajay, Akhil, Jaswant. Sickle cell anemia. The autosome linked receive triads control B, a single pair of alleles. What are the pair of alleles? HBA and HBS. These are the two types of alleles present in it. Due to this one, what happened to the sickle cell anemia? Normally, you know about the RBC look like a disc shaped, isn't it? Okay. Suppose if it is not look like a disc shaped, it look like a sickle shape due to the lack of what is that? These two alleles, some of the disorders which occurs within the blood components. Okay, it is also one of the genetically disorders. Another one is called as pedigree analysis. What is the meaning of pedigree analysis? It is an analysis of your triads in several generations of the family in humans. Pedigree analysis means what is that? What is your grandfather and grandfather, like that we are calculate means. Okay, so there is a graphical representation, look like a graphical representation as well, and other names also called as small calculations. Pedigree analysis, each one it has a specific, for example, male, it has a specific symbol, female, okay, boy or girl, like that, father, mother, sister and brother. Okay, the connection is also there is a small link, like I say, which is where in the pedigree analysis they were used graphical representation in computer when you are using the systems. Okay, for the next one. And homozygous, this is very, very important. What is by homozygous? Is nothing but the diploid conditions in which the alleles at yes given locus are identical and are also called as pure. Homozygous is nothing but the both what is that? Alleles are same. Capital T. See here, the identical factor is called as homozygous because capital T is a homozygous dominant. This is small t. It is nothing but the homozygous recessive. This is homozygous dominant. Dominant character always mentioned in the letter capital, and the recessive always mentioned in a small t. Okay, this is homozygous. Otherwise called as, it is a pure form. Both the characters is also, what is that? Capital T. This is the pure one. This one is called as, what is that? A small t. This is a recessive character, but it's pure. Okay. What about the heterozygous? See that the example heterozygous means a dominant alleles one is also present in the, and small another allele is also recessive allele is also present the same locus. This is called a heterozygous. These two one gamete present in the single locus. This is called as, what is that? Heterozygous. But which one is dominant? Capital T is the dominant. It expresses itself. That is very, very important. So the recessive character is suppressed. This is also a this condition is called as heterozygous. Zygote present the two characters, two alleles, dominant alleles as well as the recessive alleles. See here, the heterozygous or hybrid organisms containing two different alleles, both dominant and recessive. Here the both dominant and recessive, both. See here, one is dominant, another one is the recessive. For given genes, example capital T, small t present the same locus, it is considered as a heterozygous. Here homozygous means both are same, identical. This is the recessive, this is dominant. Here dominant, but both the character is present. Dominant character also present 50%, and recessive character also present 50%. And heterozygous already discussed. Different factor is called as heterozygous. This is different factors. Okay, capital T, small t, and small t, capital T. This was stamped by Batson. This stamp was given by capital T, small t, capital T. This was given by the name of the scientist Batson. Okay, so homozygous as well as heterozygous. The different factor is called as heterozygous. The stamp given by, who is that? Batson. Heterozygous stamp given by Batson. Once again, I repeat for you again and again. Okay, don't forget this. Next one is genotype. Okay, when you are going to study the genetical hybridization, monohybrid, dihybrid, that time genotype and phenotype, F1 generation, F2 generation, G generation, and also P generation. Likewise, there are 3 generations there. Okay, genotype means genetical characteristics, isn't it? Genetic constitution of an individual is called a genotype. And phenotype, already I told you, phenotype is nothing but the observable character of an organism determined by its genes. I will tell you, it is also a physical characteristics, observable characters, otherwise called as physical characters of your genes. See here, genotype is the gene complement or genetic constitution of an individual with regard to the one or more characters, irrespective of whether the genes are expressed or not. This is genotype. Genotype is nothing, genetical constitution of the individual. Phenotype, it is observable characteristics, otherwise called as physical characters. Tall is a physical characters. Dwarf is a physical characteristics. Red, white, black, these are all physical characteristics. Capital T is a dominant, is nothing but the genotype characteristics. Okay. Gene locus is the portion or region on chromosome representing a single gene. Already told you the locus, any one of the locus. Gene locus is not bad, where there is a gene present in the particular area or locus, loci. Okay. They expressed how they will be represented in a single gene. Capital T. Okay. And small t. Or capital T, small t. Likewise, these are all called as phenotype. And then F1 generation. The F1 generation, so many times they were asked, the F1 denoted as what? See here, phylyl generation. Note it down, this, my dear students. F1 generation is a first phylyl generations. F2 generation is nothing but the second phylyl generation. The 2 denoted as second. The 1 denoted as first. The first phylyl generation comprises of offspring obtained from the cross between 2 distinctively different parents. 2 distinctively, capital T, tall, small t, dwarf. These are the 2 distinctively different parents. Okay, this is the, and F2 generations. Okay, F2 generation, second phylyl generation, comprise of offspring resulting from cross of F1 generation. When we are going to cross F1 into F1, you will get F2 generation. That time, what happened? We will get the progeny. That progeny is called as F2 generation. Suppose if it is monohybrid, you will get it 1:2:1 ratio. Okay, already we studied that things also in the previous lessons. Okay, first, genetics. Will you please tell me what is meant by genetics? First of all, the introduction. Gene or the unit of heredity, already we discussed, that are arranged in a linear manner at a specific position on a specific chromosome is called as gene. So next one is the science that deals with the study of gene is called as genetics. First, what is that? There is a small explanatory aspects. The gene or the unit of heredity that are arranged in a linear manner. Note it down. This is very, very important. A specific position on the specific chromosome It is otherwise called as the science that deals with the study of gene is called as genetics. Okay, this is the another part. And the genetics also further divided by so many things according to their mode of actions. There are three divisions of genetics. One is called as transmission genetics, second one is called as molecular genetics, and third one is called as population genetics. Okay, these are the three divisions maybe exist. One is called as transmission genetics. The transmission genetics examines the principles of heredity. In this type, we are going to examine the principles of heredity. There is a specific principles also. What is that? Present in it. That things we are going to study or examine. That type of genetics is called as transmission genetics. Molecular genetics. Molecular genetics deals with the genes and the cellular process by which genetic information is transferred and expressed. What is that? It deals with the gene and the cellular process. What are the molecules? What are the genes? What are the genetic composition? What are the genetic information? Nucleotide, whether adenine or guanine, thymine, uracil, cytosine. Okay, and nucleotide, which sugar molecule present in it? Phosphoric acid, which elements? For example, it motivates the things. What are the amino acid present in it? How it will be used for the translation process, protein biosynthesis, like that, they are analyzed. Okay, so this is a molecular genetics. And population genetics records the genetics composition of a group or organism and how that composition change geographically and with the passage of time. Population genetic means what is that? The genetical characters, it's also transferred from the one generation to another generation. So what are the changes which occurs? What are the changes occurs in the genetical aspects? Or what are the new genes present in it? How? Composition of changes. This is very, very important. Geographical and also with the passage of time. It is also chronological order. Okay, we are all human beings, isn't it? Our ancestor is, what is that? Our ancestor is monkey, isn't it? Ape. Okay, so already we discussed that things also, isn't it? Okay, go to the next slide. So model genetic organism. We were used so many genetic organism for the identification in and new innovation discovery aspects. We were used for the biotechnology aspect and also molecular biology, genetic engineering, gene technology, and so many things. Is that there are 6 model organisms that have been that subjected or intensive genetic study or as follows. We were used, okay, genetical studies, the laboratory aspects. See, first one is called as Drosophila melanogaster. It is otherwise called as a fruit fly. Fruit flies. Note it down. This is very, very important. This is called as one of the fruit flies, Drosophila melanogaster. Escherichia coli, already you know about E. coli, mostly used for the biotechnology aspect. When you are going to produce humulin and other and plasmids oriented, when we are going to study that genetical aspect, resistant characteristics, when you are going to study the plasmids and their characteristics, we can use it E. coli laboratory aspect. And another one is called as, what is that? Caenorhabditis elegans, a nematode worm. Nematode worm also sometimes they were used for the model genetic organisms. Arabidopsis thalaina. This is also another one. Mus musculus is another one. And the last one is called as Saccharomyces cerevisiae. Bakery yeast. The zoological name is also so many times they were asked which one, the yeast. The zoological name is called as Saccharomyces cerevisiae. It is obtained from the sugarcane molasses. Sugarcane molasses. This is very, very important. These are the 6 model organisms that were used for the genetical studies. Okay, this is very, very important one. What are they? Drosophila melanogaster, E. coli, and Canorrhoea habitus elegans, Arabidopsis thaliana, musculus, Saccharomyces cerevisiae or cerevisiae, bakery yeast. Bakery yeast is otherwise called as Saccharomyces cerevisiae. This is also asked in 2017 or 18, I think. Okay, just go and check it. You will get it. Where will you get it? Saccharomyces cerevisiae, also get it from the sugar cane molasses during the preparation of ethyl alcohol or sugar, table sugar. Okay, sucrose, that things. Okay, go to the next one, my dear students. See, other species that are frequently the subject of the genetic research and considered genetic model included. This is also so many, what is that, times they were used. This is the biotechnology aspects. Neurospora crassa, note it down, Neurospora, one gene, one enzyme hypothesis was done by the one of the fungi is called as Neurospora. Note it down, my dear students, Neurospora, fungi. One gene, one enzyme hypothesis, pedilentatum. Note it down. And Giamase already in our but Danio rario, zebrafish. The zebrafish otherwise called as Danio rario. And Xenophus lievis, cloud frog. These are the other name. So cloud frog is sometimes they may call us, what is that? The zoological name is called as, what is that? Xenophus levis. And zebrafish, Danio rero. So these are the, what is that? Other species that are frequently the subject of the genetic research purpose. Genetic research purpose they were used. Okay, so go to the next slide. The earlier concept of the hereditary, here you must note it down. First, who concept, who proposed the concept, and correct or incorrect, likewise. Early stage. Okay, already we studied it. What is my nomen ambiguance? Binomial nomenclature already in our report, isn't it? Any one of the plant or animal, we are giving 2 words or 2 names is called as binomial. This was proposed by the name of the scientist called as Carolus Linnaeus, isn't it? The Carolus Linnaeus stated the statement binomial nomenclature, all the plants, is it correct or not? Some of them are wrongly given, isn't it? That was corrected by some of the other scientists. This is called as author citation already in our book. What is my author citation? When you are going to study the taxonomy, the biological classifications, that time you know very well about. Otherwise, we will discuss later. Okay, when you are going to study the lesson. Here, the early concept of hereditary, they have given each one, so many scientists, they have given their own explanation. See the concept, first one, spongenesis. This was given by the name of the scientist called as by Charles Darwin. Spongenesis. What he proposed, you know, genetic information travels from different part of the body to reproductive organ only. Okay, what is the statement? Genetic information travels from different part of the body to reproductive organs only. This is correct or not? This is the incorrect statement because gene is responsible. Okay, genetic information travel, they told, from different part of the body to reproductive organ only. It's not a right reproductive. Suppose if it is changed here, a chromosome like that, you know, it is a correct. Okay, next one is inheritance of acquired character. What is meant by inheritance of acquired character? Already having the character, it's also transfer from, what is that? Another generations. This statement was given by the name of the scientist by Lamarck. Lamarckism, already we studied. acquired triads become incorporated into the hereditary. What is that? Hereditary. Okay, so informations, hereditary information. This statement is also incorrect. Okay, so this is also another one. And then preformatinism. Preformatinism. Miniature organism resides in sex cells and all triads are inherited from one parent. This is also incorrect because the sex cells already in order, both the triads present in it. It do not comes from the single parent. Okay, this is also a incorrect one. Blending inheritance, blending, gene blend and mix. Okay, blend. Okay, but it will not blend because it can segregate. When it heterozygous condition, they can segregate itself and separately. So this is also a incorrect statement given by nobody else here. Germ plasm theory, note it down, my dear student. This is very, very, very important one. So many times they were asked the question. This germ plasm theory was given by the name of the scientist called as Augustus Weismann. Augustus Weismann. See here, all the cells contain a complete set of genetic information. This is the correct statement given by the name of the scientist called as Augustus Weismann. Okay, so all cells contain a complete set of genetic information. This was the correct one. Cell theory. The cell theory was proposed by the name of the scientist called as Sladen and Swan. Sladen is botany, Swan is a zoology. So many times they were asked the questions. Sladen theory, a cell theory was enhanced by the which one of the following scientists? Cell theory was proposed by Sladen and Swan. Sladen botany cell were observed, particularly pork. Okay, an onion cell like that. And then Swan is also observed. Chick embryo, I think. Okay, that thing is also observed by the scientists. But the cell theory was enhanced by who, Swan, not Sladen. Okay, see here. All life is composed of cells and cells arises only from the cells. This is already in our all living organism made up of cells. Each cells arises from the pre-existing cell. That is called a cell theory. This is the correct one because we are all comes from our parent. Our parent also comes from their grandparents. Grandparents, grandparents. Likewise, because each and every cells also arises from the pre-existing cells already present in it. That cells also segregate the another cell, another generation. It is also a correct one. And Mendelian inheritance, another one. The triads are inherited according to the, what is that? See that one? Defined principles. The Mendelian inheritance triads are inherited according to the defined principle. It is also one of the correct. And Mendelism, already you know about. The Mendelism, there are three things that we are going to study. Law of dominance, law of segregation, law of independent assortment. That things we are going to study in, what is that? Mendelian inheritance. Next one. We are going to study the explanation of the G.J. Mendel in the year 1822 and 84. Who is the father of genetics? Who is the father of genetics? G.J. Mendel is the father of genetics. Historical aspects, sometimes they were asked the questions. Okay, where he was born. Okay, his experiment was how many characters is there? Why he has chosen, which one you know? Sweet pea, otherwise called as Pisum sativum, as a test plant. Like that, they are asked the question. So Mendel was born in the small village of the Heidelberg in the Israel, Austria. The Mendel was born in a small village, Austria. Note it down. This is very, very important, which is not part of the Czech Republic. He conducted the breeding experiment from 1856. Sometimes they may ask the question, how many years they conduct the experiment? Okay, 1856 to 1863, and presented his result publicly at a meeting in Brno. Okay, so this is this year. Okay, 1856. Don't forget 1863. How many years here? Just imagine here, 4 plus 3, 7 years. Mendel selected 7 visible characteristics, each with 2 contrasting triads. See here, the how many years is there? 7 years, isn't it? How many characters also chosen by the GJ? 7. Okay. Contrasting character. What does it mean by contrasting? Tall is a opposite of dwarf. White, another one is called as black. Wrinkle, round. Okay. Yellow, red. Likewise. He also kept an accurate record of his experiment, giving all the details of numbers and type of individual which are the necessity in genetic studies, because then only we are studying. Otherwise, what happened now? We will not study this genetic. Okay. So the father of genetics, who is that father of genetics? G.J. Mendel is the father of genetics. He explained how triads are transmitted from one generation to the next generation. He only explained how triads are transmitted, how the characters are transmitted from the one generation to another generation. Okay. The result of his experiment were published in the Science Journal. Note it down. This is very, very important. Natural History Society of Brno in 1866. He died in 1884 of kidney disorders. Sometimes they may ask the question, which years? The Mendel chosen Gordon P. Otherwise, the botanical name is called as what is in Pisum sativum, civet P. Gordon P. For his experiment for the following reason. Why he has chosen for this one? Sometimes they may ask the questions. Why the Gordon P were chosen by the Mendels? Why? See here. Anybody else? This one? Anybody else, my dear students? Anybody? Ajay, Jaswant, Ritika, Agil, Dhanalakshmi. Anybody else? Okay. The bee plant is a small, easy to grow and can be cross-breed artificially. The bee plant is small, easy to grow and can be cross-breeding artificially. This is the first point. Second point, it is a bisexual, a male and female flower present the single flower. And so it is self-fertilizes in nature, but can be easily cross-pollinated experimentally. This is another point. Nature is possible. At the same time, cross-pollination is also experimentally possible. That is a second reason. Third one is it can be reproduced a large number of offsprings. This is also another one. Number of offspring is also very high. Okay, during the hybridization process, otherwise called as cross-pollination. It can complete its life cycle in one season. One season means maybe take 3 to 4 months. So per year, we can, what is a hybrid? Many times, 4 times. This is, suppose if it is 3 months, means what happened? We can breed 4 times per year. Okay, see that. The last one, many true breeding varieties of bees are available. True breeding. This is also important. That's why this was chosen by the plant was chosen by the scientist. C.J. Mendel was chosen. Okay. See here, this is the plant. This is a tall plant. They denote it as a capital T, capital T. This is a small, otherwise called as a dwarf plant. It is also small t, small t. This is the contrasting character. Tall is a dominant character, and opposite contrasting character is the dwarf. So it is a small one, this is a large one. They denoted the letters called as capital T, capital T. This one is called as small t, small t. These two heterozygous condition was denoted or named by the Batson. And you know about that things also, isn't it? Okay. Mendel studied the following 7 characters with the contrasting types. So what are the characters? How many characters is there? Already we discussed 7 characters there. That 7 characters, what are they? First one is stem height. Already told you stem height. Height is denoted as tall. Another one is contrasting character is dwarf. Large or small. This is the first characteristics. Second one is seed shape, round and wrinkled. This is another one. Okay, seed shape, round and wrinkled, contrasting characters. Seed color, yellow and green. This is also a contrasting character. Depends upon the seed color. Pod color, green and yellow. This is another point. Here the seed color is yellow and green. Here pod color is green and yellow. Opposite. Okay. And pod shape, inflated and constricted, shrinkage like that. It comes. And flower colors, violet and white. Flower position, axil and terminal. These are the 7 characters were chosen by the Mendel. C.J. Mendel. Okay, my dear students. Okay, these are the things. Okay, see here another tabulation has given here. The character is first one is called as plant height, tall and dwarf. It is depends upon their chromosomal aspect they have been given. Sometimes they may ask the question plant height, tall and dwarf. How many chromosome number present in it? 4. Okay. Flower and pod position, axil and terminal. Flower, you know what terminal means? The tip of the flower. Axil means you know what but stem and leaf. In between the stem and leaf, there is a ax. That axil position is also there. So this is also a 4 chromosome number. Pod color, already we studied green and yellow, 5. Pod shape, inflated and constricted, and 4. Color of the flower, violet and white. This is also 1. Color of the flower. When we are going to discuss the color of the flower, how many chromosomes present in? Only 1 chromosome present in it here. Seed shape, round and wrinkled, 7. This is very, very important one. 7 chromosome is also present in it. Seed shape, round and wrinkled. How many chromosome number is there? 7. And last but not least, the seed color, which one you know, yellow and green. This is also a 1. Okay. Here, how many 4 is there? 3, 4 is there. What are they? Plant height, flower and pod position, and pod shape. Okay. So pod color is 5, and the color of the flower is 1, and seed color is also 1. Seed shape only, how many chromosome present in no? 7 chromosome present in it. Okay, my dear students. Okay. So that's all about today classes. We'll see the remaining on the next classes. What are the things we already studied? Please summarize the things. So already know about what is meant by genes, inheritance of characters. Alternative form of genes is called as alleles, autosomes, and what is by autosome, what is by allosomes, how many pairs present in it, what is by factors, and linkage, what is by linkage, what is by Punnett square. Punnett square is nothing but the graphical representation of a calculated probability of all possible genotype of spring and genetic crosses. And incomplete dominant, which plant they were used for incomplete dominant, Merophilus jalafa, otherwise called as 4 o'clock plant. And mutation is also what is by sudden inheritable changes. Okay, so this is important. So we will see the remaining on the next class. Bye, take care.
Children, after 5 minutes, join again for biology class.